कम करना भी है उस पे भी बिना रेडी बहुत मजा है मेरे को वो इंग्लिश सर जो स्टार्टिंग में बहुत अच्छा लगा एक सर ने इंग्लिश स्टार्टिंग गाना मेरा भी बहुत अच्छा लगा मतलब हमारी इसमें कर रहे हैं मेरे को मेरे भाई से पूछना है मेरी बहन भी खेलती मतलब पसंद या बीच में करते हैं साइलेंट तेरे पास कौन से हैं? हाँ साइकिल से पता है जब हम लोग 50-50 रुपये तुझे तीन बार अच्छा था ये दो बजट मतलब पर पर्सन लेगा बजट इंक्रीज कर अच्छा था चॉकलेट मोमोज करके कुछ तो था वो नाम से मुझे पसंद नहीं आता नहीं क्या पड़ेगा टेबल का नीचे बनाना पड़ता है ना पनीर सब मशरूम मैंने सिर्फ एक बार मेरे पास बहुत ज्यादा है 900-800 इसके लिए ना अगर आप लंच के लिए जाओ तो नाम का खाना कुछ दिन का मिलता है ब्रेकफास्ट में बहुत स्लोवर्ड भी है उसके बाद स्लोवर्ड महंगा है क्या कितने का पर पर्सन से उसके बाद स्लोवर्ड कितना टाइम स्लोवर्ड पार्टिकुलेशन फिर भी स्लोवर्ड से मिल रहा है पार्टिकुलेशन बहुत मस्त होता है तो ये और ट्राई कुछ खाया ऐसे खड़े होने में पैर दिखता है इनको आईडी है तेरे पास वो आईडी नहीं चलेगा वो वही चलेगा वरना कुछ नहीं होगा वो आदमी पे उसको आईडी तेरे पास ये नहीं चलता प्लीज कैमरा में आ सकते हैं तो वो आईडी खरीद दिखा था अरे उसका अलग नहीं वो तो आईडी ना आईडी किससे लिया था पुष्पा
You complete the second question. I'll give you 1 minute. 1 minutes में हो जाएगा. चलो करो, complete करो. Right, you finished? Any doubt in the questions?
Not understanding C.
Not understanding C. What is that? Maybe. Okay, what's the setup? Process of osmosis, 3 potato cubes, 3 different beakers, 3 different solutions. Okay. 24 hours you have to observe in the first beaker the potato cube increases in size. In the second beaker the potato cube decreases, and in the third beaker no change. The following diagram shows the result of experiments. Give the technical term of the solution used in 1, 2 and 3. 1 का क्या होगा? Hypotonic. 2 का क्या होगा? Hyper. 3 होगा hypotonic. Okay. In beaker 3, the size of the potato cube remains the same. Explain the reason.
The concentration of solute and water is almost same.
Concentration of the solute and the water is the same on either side. Concentration of solute and water is the same in the beaker and inside the potato cube. In the beaker and inside the potato. Remember, whenever you are talking about the concentrations in terms of osmosis, you are always to compare relatively or to write. C. Write the specific feature of the cell sap of the root hairs which help in the absorption of water. Hypertonicity. Remember I explained you that write the specific feature of the cell sap of the root hairs. Cell sap of the root hairs are always hypertonic. They are always hypertonic as compared to the surrounding soil water. As compared to surrounding soil water. Hypertonic as compared to surrounding soil water. What is osmosis? That you know. Number 5. How does a cell wall and a cell membrane differ in their permeability?
Cell wall cannot expand, cell membrane can expand.
No, that is not your source, right?
Cell wall is freely
Cell wall is freely permeable, cell membrane is selectively permeable. Done. Next what? Next we are to understand the experiments let us see. This is a very important section of the chapter. One question of this type is almost every year they will come up. So you want to understand the different setups, analysis, the different analysis, the give reasons, the sorting up. So I will do quite a number of them. That should be enough for understanding. But anyways, if there is anything doubtful, you should always raise your doubts. Let's start with the first concept. So what am I talking about is the experiments. I'll start with the first experiment. The first experimental setup. Now what is the first experimental setup? There is a beaker and there is a thistle cup. The first setup that I'm going to discuss would be a setup something like this, where this is the thistle funnel. Thistle funnel. This is known as the thistle funnel, and this is the beaker. We know this. So in this setup, you can very well see that there is a liquid or solution inside the thistle funnel, and the opening, the mouth of the thistle funnel is fastened or closed with the membrane. Now this can be the cellophane or the parchment, anything. This can be the cellophane or the parchment paper. Now, what are we supposed to analyze? First of all, in the first setup, try to understand. I'm talking about water here, and I'm talking about a sugar solution. Try to always follow the rules and the concept. Okay. So I'm talking about the first setup says beaker almost half filled with water, thistle funnel placed in such a way that the mouth of the funnel is almost on the bottom of the beaker, and the mouth of the funnel should be tightened with a semi-permeable membrane. Any of these. Then what? The next one. Mark the initial level of the solution where in the thistle funnel. So I mark it as the initial level. And mark the initial level of the water in the beaker. So I mark the initial level, and I have to wait for a particular period of time. I wait for almost about 4 to 6 hours. That's the minimum time period that we generally take for this type of experiment. It can be even for a day. But always to understand, in any of these, as compared to this type of experimental setup, and even in case of the rules, the movement is not very fast. The movement is slow. The movement is slow. So let us start with this. So in this first setup, you get to see all these. Now, what are you expecting after a particular period of time, experimental period of time? Say suppose I normally give a time period of something around 8 to 10 hours, or it can be quite a number of time, 12 to 14 hours also, because the movement is quite slow. That always you have to keep in mind. Achha. Now, In this type of a setup, what happens? After this experimental period of time, it is generally seen that the level of the water keeps on rising. The level of the water is now rising. Is it a very remarkable rise? No, not a very remarkable rise, but yes, there's a rise. So now this becomes my final level of the water. Should the level of the water in the beaker remain the same? No, there will be a drop. There will be a drop, and this will be the final level of the water in the beaker. But what is the reason? The reason is this is a dilute medium, this is a concentrated medium. Anyway, this is water. Even if I talk about 2% sugar solution, even if I'm talking about 2% sugar solution, then also it's a concentrated medium as compared to this. So the water is moving. Water molecules are moving from the beaker to the funnel or funnel to the beaker. यहाँ पे देखो, यहाँ पे कुछ नहीं है, सब यहाँ पे है. Water is moving from funnel to beaker or beaker to funnel. In this situation, I'm sorry, in this situation, beaker to funnel. आया समझ में? I'll repeat if you're not clear. Water है beaker में. I did not talk about a solution. It's just water. Funnel है, funnel के अंदर solution है. जो भी है, 1% है, 2% है, 10% है, 50%, whatever. It's a solution. The strength of the solution is not given. फिर भी, you have to understand, this is a concentrated medium. The moment I say concentrated medium, what do you understand? Water is more or less? Less. Water is less. यहाँ से water is less here, no? So what should be the direction of movement of the water? Beaker to funnel or funnel to beaker? Beaker to funnel. Okay? So the direction of movement of the water would be this way. Now I change the parameters. I write same head. Initially I said it was 2% sugar solution. But now I say that it is not 2%. It is somewhere around, say suppose, 20% sugar solution. Then what will you observe in the level of the solution of the funnel? What will be the change in the level of the solution of the funnel? If the setup is something this way, the funnel has 20% sugar solution, outside in the beaker is what? What will be the change? Observation क्या होगा?
The water level will rise even higher.
The water level will rise, matlab that's another experimental setup that I'm talking about. That's another experimental setup that I'm talking about. So after this experimental period of time, what should I show? What will be the experimental observation? क्या होगा?
Rise in water.
Again, no? Again there will be a rise in the level of the water. क्यों? Obviously this is more concentrated, this is more dilute. So this much you understood. Now let us talk about another change. Now here it is 20% sugar solution and here it is 10% sugar solution. I am not taking water now in the beaker, but I am taking 10% sugar solution. I am taking 10% sugar solution. I am taking 10% sugar solution and inside is 20% sugar solution. Now what will be the observation?
Rise in water level and
In the funnel. आया समझ में? First condition में भी ये dilute medium था. Second condition में भी ये dilute है. अभी भी ये dilute medium ही है अभी भी no, because this is a 10% sugar solution. 10 parts solute, 90 parts water. Water is more here. 20 parts solute, 80 parts water. Water is less here. So direction of movement of water will be still this. Still this. Still this. Water will still move from beaker to funnel. Water will still move from beaker to funnel. But now, if I say this is the change, then
water level will decrease in the funnel and beaker's water level will rise.
Increase. The water level will decrease in the funnel आ रहा है समझ में? And the water level will increase in the funnel. But my question is, so now in this case the direction of the movement of the water is not this but this one. After 12 hours, when I taste this water, is this water sweet? Is this water sweet?
Yes.
20% sugar solution है, water इस तरफ move हो रहा है. Is this solution sweeter? Meaning, is it sweeter than before?
Yes.
No, I don't want this sweet.
क्यों?
Because only water is good.
Correct. This is a semi-permeable membrane. It will allow the selective movement of molecules. Water, not sugar. ये sugar का movement को allow नहीं करेगा. It is only osmosis we are learning. We are not learning diffusion. This is osmosis. It will only allow the movement of the water, not the movement of the solute, not the movement of the sugar. So water will moving and the level of the solution in the beaker is increasing, but gradually this solution in the beaker will become faint and faint sweet. It will not become gradually sweeter. आ रहा है समझ में? So this is one. Now we have to understand some other concept, something like that. Okay. Now I'm not going to use semi-permeable membranes. Now, maybe we use one muslin cloth. Muslin cloth we use. Here, in this setup, चलो ये setup को थोड़ा change करेंगे. Let this be concentrated inside, and let this be water. Let this be water like before. Then, in this case, what are we trying to understand is, वही पहले जैसे setup, water inside a sugar solution and a membrane to be fixed at the mouth of the funnel, but now we are not fixing parchment or cellophane, but we are attaching a muslin cloth. Muslin cloth, if I give you a fair idea, how should it look like? Muslin cloth basically is just like those fine pure cotton cloth. You have seen, no? Pure cotton cloth where the holes are quite big. So it's something like that. The muslin cloth that we understand. Muslin cloth fabrics are also there. No? So now this muslin cloth, when we talk about, its nature is it is freely permeable. Muslin cloth is freely permeable. Earlier we were talking about semi-permeable. Now I write. When I took this muslin, what is the nature? I am aware that this is freely permeable. Now you answer these questions. Then, after the experimental period of time, what will happen to the level of water in the beaker?
It will decrease.
Level of water in यहाँ पे तो वो solution है नहीं 20% sugar solution as it is है पहले जैसे. And I'm saying that now I'm not fixing cellophane, I'm fixing muslin cloth which is freely permeable. So after this particular period of time, what should happen to the initial level of the water in the beaker? What should happen to the initial level of water in the funnel? Freely permeable. ये याद रखना है. सोचो. Freely permeable अगर है, what should happen to the level of solution here? What should happen to the level of solution?
Water level will rise.
Water level will
Rise.
water level will
Rise in the funnel.
rise in the funnel. Okay. And
Water level decrease in the beaker.
since it is freely permeable, we also come across that this is sugar solution, whatever the strength is, and this is water. So yes, the water level in the funnel will rise gradually. It is freely permeable. And now, unlike the previous cases where I said the level of the water in the beaker will rise but very slowly, no, I said because it's the cellophane. But with muslin, the level of water in the beaker will rise very sharply, and the level of solution in the funnel will drop also very sharply, meaning you can easily understand the difference. Now, will this water which has increased, the level of the water which is increased, is it going to be sweet?
Yes.
Is this water is going to be sweet?
Yes.
Q.
Now because even the sugar will get transported.
Correct, because now we are talking about freely permeable. So of course this water is going in, but sugar molecules are also coming out. No? The sugar molecules are also coming out. Some sugar, some water will also come out. Some water will also come out because water is more here. So the majority of the water will go this direction. But sugar is also moving in this direction. Both ways of movement is happening. So abruptly we get to see that the level of the solution in the beaker is increasing, funnel is dropping, and this solution is gradually becoming sweeter. This solution is gradually becoming sweeter. Then instead of muslin, now I take something else. Instead of muslin, let us take a rubber sheet. What is the property of the rubber sheet? Impermeable. What is the meaning of impermeable?
It won't let any
Does not allow the transfer. Impermeable. So now the rubber sheet we are taking. Then what will be the difference, Bolo?
There will be no difference. It will be
It will be the same.
Rubber sheet, it will be the same meaning what?
the water levels in the beaker and funnel will be same.
This is the rubber sheet. Is there any change in the level of the water in the beaker? No. Is there any change in the level of the water in the funnel? No. Why? Because the rubber sheet doesn't allow the movement of the water. Does it allow the movement of the solute? No. Is the beaker water sweet? No. So nothing, no observation we generally see here. So instead of the rubber sheet, we can also take a plastic sheet. Same result. Result with observation will be the same. So this is the 4 experiments that we talked about. The modification of the experiments in different aspects and what to analyze from each of those experiments. What do you analyze? What do you understand from each of these experiments? So similar experimental setup, let us also try to understand here. Almost same like this when we talk about the whisking bag. Almost the same thing, whisking bag. And here we are talking about this. And now here, this and this, what is the difference? The first experiment that we were talking about, the cellophane here, no? So this is also almost the same when I talk about the use of cellophane. And now we talk about the whisking bag, almost the same thing. What's happening? Here what do we take is, inside the whisking bag, we now take the solution. How? Inside the whisking bag, we have to take the solution. So we take the beaker this way, and then in the beaker, what are we to place? We are to place the water, exactly same like the previous. And then there will be a glass tube, and then we have got the whisking bag attached here. We have got the whisking bag attached here. So this is the whisking bag. But if we take outside in the beaker, we have taken water. And this is the whisking bag. And what did we take inside the whisking bag? Inside the whisking bag, we have taken a solution. What will be the strength? The strength is generally not shown. That can be 10%, 20% sugar solution, salt solution, anything. Now this is there, and here, after a particular period of time, what do we get to observe is that the level of the solution in this tube increases. So in this case, whenever we are talking about that, if the whisking bag, if the setup is something this way, where there is a solution inside, water outside, what is the directional movement of the water? This way. But to understand, the whisking bag is also a semi-permeable membrane. This is also acting as semi-permeable. This is also acting as semi-permeable. So that is the reason the water level is rising up in the tube. Can you give me a condition when the whisking bag will start shrinking? Under what condition will the whisking bag start shrinking? What should be the change in the setup to show that the whisking bag is shrinking?
The concentration in the water in the beaker will be higher.
So यहाँ पे if we take a high concentrated sugar solution and inside it we give water, or here if I keep 20% sugar solution and here if I give 5% sugar solution, then also the same observation we will get, where the water will be lost from the from the bag. Water will be lost from the bag. Water will be lost from the bag. Water will be lost from the bag. When there is outward, now if I consider this is a cell, when there is outward movement of water, what is that called? Exosmosis. So under what condition exosmosis happens? When the external environment is concentrated or dilute? Exosmosis, when does it happen? When the external environment is concentrated or dilute? Concentrated. And when does endosmosis happen? When the external environment is dilute. Okay? Now, you will start answering these questions. Chalo, first, are you able to read? Are you able to read? Chalo, I'm just making this a little bigger. Are you able to read? I want you to answer these questions in your notebook. Notebook में लिखना है ना. You may take the reference of this figure. Figure draw नहीं करना है. You may just write down answer number 1. What is the figure in your this one textbook? See the first experiment. 4.4. हाँ तो reference दे दो. Diagram reference figure 4.4. Text का reference 4.4. You don't have to draw the diagram. Just see the diagram and answer the question. Later if you see that कौन से diagram पे हमने ये question किया था, so only for that. Reference of the diagram is figure 4.4 text. Now you see the figure from here and read the questions. I'll give you only 5 minutes per question, not more than that. In the notebook. Notebook में लिख के करना है, so that you have the entire set of questions answered in your notebook. Some similar type of questions are there in your exercise texts also that we will do later. पहले ये करो. Should I read out?
Like which paper is it?
Which one?
Which paper is it? Please and
Parchment paper. Parchment paper is there. The base, it's titled with the parchment paper. Any confusion, questions not understood, clarify. Answer should be very short, very brief, one word, one sentence, no description. Huh? Done? Fifth answer. The answer for the fifth and the fourth are related. Answer for the fourth and the fifth are related. Now, if you know the answer for the fourth, you know the answer for the fifth also. Done. Done. You try this. What is the third question? What is the reason? What I straight away moves from a region. That's not a movement of the solid. So if that's the reason, we have to justify that yes, it is only allowing the movement of water. Right, neat, presentation very good. That's why I'm giving you enough written work so that you know how to write down. All done? All done? Yes. We will increase this. You can also write fall and rise. These are also other words that we use. Huh? No. Here.
Outside only water is open.
Okay. Done? So she has written for 10 mark answers in this. Huh? इतना क्या लिखना है? In science, पूरा this is not English and grammar. पूरा sentence नहीं लिखोगे तो भी full marks मिलेगा. ये word खाली लिखोगे ना, then also you will get full marks. Unnecessary, this is waste of time. ये तो English comprehension नहीं है, ये पूरा sentence लिखना है. You can save time on this. For biology, for science, you don't have to be number one grammatically correct. Now, I'm not saying that you should be wrong. And standards, good student, good schools, of course you should not write wrong English, that is different. But stress on what? Stress on the key terms. He had used the word endosmosis. He used for it. You have to use the key terms, endosmosis, exosmosis, जो भी chapter, जो भी subject. I'm teaching biology. Biology के कौन से key terms है? Your answer should be loaded with key terms. The cell shows inward movement of cell shows endosmosis. Not that generally a big sentence. Cut short that sentence and give one word. So that way you should learn how to get. Just one word. This type of answers are just one word answer. पूरा sentence नहीं लिखोगे तो भी कुछ फर्क नहीं पड़ेगा. The correct word, the correct key term. That's it. Reasonable observation. No. इतना लिखने का जरूरत नहीं. Only this no was enough. Why? What is the reason? How will you justify? You will only justify by writing the partner is not allowing the movement of the solid. Partner is not allowing the movement of the sugar. It's only allowing the movement of water. Done? Huh? In the fifth one. फर्स्ट में क्या लिखना है? What is the answer for the first? What will happen to the level of the water in the beaker?
It increases.
It
Increase.
increases. Next क्या होगा?
It will increase now.
What will happen to the level of the water in the? अच्छा, no, level of the water. We are talking about water solution inside. No, solution inside. So if it is solution inside, the level of the water in the beaker will drop, decrease. Solution of the water in the beaker will drop. Next बोलो. Correct करो. पहले तुम्हारा आंसर correct करो. Solution of the water in the beaker will drop because inside it is solution, outside it is water. Solution level of the water in the beaker will drop. Next बोलो. Now with that reference keep saying. Second क्या होगा? What will happen to the level of the water in the funnel? Funnel में solution है, beaker में water है. So if it is water outside in the beaker,
It will go in the funnel.
and beaker का
Sugars.
level funnel में अंदर जा रहा है, so the level of the beaker is dropping, decreasing,
And funnels increase.
rise होगा. Done. अब पूरा महाभारत गलत हो गया. Make a habit of writing very short answers. In all this set of questions, I'm going to give you a lot of practice. पूरा sentence नहीं लिखोगे तो भी it's okay. Again, this is not English comprehension language नहीं है कि full sentence पूरा full stop से लेके comma तक लिख. No need. एक word, just that word, enough. आया समझ में? That's the way of answering this type of question. पूरा sentence नहीं लिखने का जरूरत है. यहाँ पे I would answer writing that this will decrease, this will increase. No, yes, full 5 marks will get. Understood? So see how much you have to write. If there is a need of writing a sentence, then of course you have to write. For definition, you have to write that full sentence. No, then you have to write on the full sentence. चलो number 3 का क्या लिखना है? What is the reason? Water level beaker में decreased, funnel में increase. Reason क्या है? Reason क्या लिखोगे? What did we learn all these experiments? We have almost 6 experiments we studied. What did we learn in all these?
That all the solutions go into the beaker.
Water always moves from a region of high concentration to low concentration. यही substance हमने सीखा है in all these experiments, नहीं? So what is the reason for the observation? Reason of the observation is water is moving from a region of low concentration to a region of high concentration. यही हो रहा था. That is the reason, no? यही reason है कि नहीं?
Yes.
Water is moving from a region of high concentration to a region of low concentration. That is one sentence that we read. Number 4. Will there be any change in the taste of the water of the beaker?
Yes.
सोच के बताओ. एक और बार सोचो.
Because it will be more sweet. Water.
अभी यहाँ पे है solution, यहाँ पे है water, and अभी हमने क्या बोला था कि the water will move this way. The water is moving. करते करते this water is dropping down, and this solution is rising up. अब मेरा क्वेश्चन ये है कि ये जो बीकर में जो वाटर बचा है, will that be sweet? कोई स्वीटनेस हम एक्सपेक्ट कर सकते हैं? नो. समझ के बोलो. आंसर समझ के बोलो. Why no? Now the answer, 5th question is why no? Why no? Give a reason. Why no?
As the water from the beaker had moved into the funnel, so there was no sweetness in the water before.
No, but my question is ये बीकर का जो water बचता है, will that be ever sweet? क्यों?
Because the sugar solution did not move into the water went into the funnel.
हाँ, तो sugar solution, why did not it move into the beaker?
Because of it had the high concentration.
नहीं, because the semi-permeable membrane will only allow the movement of the water. Sugar molecules तो movement allow ही नहीं करेगा. Sugar molecules का movement allow करेगा? No. तो कौन सा membrane just now we study कि would have allowed the movement of sugar molecules also? muslin cloth. We have learned, no, that had it been the muslin cloth, then it would have allowed the movement of sugar also. तब जाके beaker का water would have been sweet also. So that is what you have to write. How can you justify that the parchment is selectively permeable? Because the parchment is allowing only the movement of water, not sugar. Parchment is allowing only the movement of sugar, not water. फटाफट एक और करो. Still now some of you are not clear with the concept. Right. Now what I told you, you will answer in just one word if needed. Sentence लिखना है तो लिखोगे. अगर sentence नहीं frame करना है तो चाहिए नहीं. एक-एक word पे अगर answer है तो एक-एक word पे लिखोगे answer. Key terms चाहिए तो key terms लिखोगे. Make a habit of using the key terms as much as possible. Endosmosis, exosmosis, tonicity, etc. Hypotonicity, hypotonicity. जितना key terms लिखा है उसको use करने की कोशिश करो. अच्छा, in this experiment दिखाई दे रहा है. I will tell you. यहाँ पे क्या है? Same beaker has water. Funnel has sugar solution. वही same setup and semi-permeable membrane, but the set of questions are different. What is the first question? Name a semi-permeable membrane. Any. Name a semi-permeable membrane. Likho. Number 2. What happens to the level of the liquid in the funnel if the beaker contains hypotonic solution? अभी यहाँ पे तो water दिखाई दे रहा है, but I am saying ये water को substitute करके एक hypotonic solution अगर मैंने ले लिया, अभी तुमको क्या समझ में आना चाहिए कि funnel has a solution, beaker में भी हम we will take another solution which should be dilute than the funnel, hypotonic मैंने बोला. If the beaker, if we substitute the water of the beaker by a hypotonic solution, मतलब immediately तुमको you should be able to judge the solution in the funnel is there. I have to take a solution less dilute in the beaker. Beaker वाला solution dilute होना चाहिए, funnel का solution concentrated रहना चाहिए. आया समझ में? With that thought, keep. अरे don't sneeze like this. You are increasing my fear now. As it is, I am not able to take out my mask. आधे लोग आजकल mask नहीं पहनते. I am still wearing it. Because all of us we are wearing it. And this person is sneezing now. चलो. ये सोचो कि अभी ये water नहीं है, but it is a dilute solution, dilute than the funnel. फटाफट. 5 minutes से ज़्यादा नहीं दूँगी. एक-एक word चाहिए तो एक-एक word ही लिखना है. Think and write. Understand and write. कहीं पे कुछ भी डाउट है प्लीज आस्क, एनीथिंग यू फाइंड कंफ्यूजिंग। Done? I'm sorry, make a correction here. Huh? Number 4 देखो. Will there be any change in the taste of the water? No, it should be, will there be any change in the taste of the solution of the liquid? Because we have already substituted the water by a hypotonic solution. Okay? Will there be a change in the taste of the solution of the liquor? Will there be a change in the taste of the solution of the liquor? Done? Let me see what you put. Very good. Clear now? Any type of question on this? The diagram will remain the same, the setup will remain the same, but you have to read the question. What is the change in the experimental setup? And based on that, you have to answer the questions. I done.
There's the hypothetical solution. We only limit to that question of the full thing we have to take that.
That only you have to take. That's what I said, no? इसीलिए ये fourth में I forgot to change this word. Water के जगह यहाँ पे hypotonic solution ही लेना है. अरे तुम लोग कितना लिखते हो बाप रे? हाँ? हिस्ट्री ज्योग्राफी कैसे कंप्लीट करोगे पेपर? आधा पेपर तुम लोग अभी तो कंप्लीट नहीं होगा. कितना लिखना है वो सब सब्जेक्ट्स में, इसमें तो लिखना ही नहीं है कुछ, यस नो हो गया, आई डन दो देखते हैं व्हाट डिड यू राइट। नहीं, नंबर 3 में सेमी परमेबल की प्रॉपर्टी नहीं लिखना है। व्हाट इज द रीजन? Beaker has hypotonic, I said. So the moment you know the word hypotonic, I said no, हमेशा compare करना है. So the beaker has hypotonic. So you understand that the funnel has concentrated. So water will always move from dilute to concentrated medium. ये reason लिखना है. अच्छा, number 5. The best answer that you should write is. I asked you. When will osmosis stop? When will osmosis stop? What do we understand? Say suppose I talk about an RBC cell in our body, I talk about the plant cells, or I talk about two medium separated, I give you that question also 5%, 10% sugar solution. So, potato cubes, anything. When will the movement of the water stop? That's a basic concept of osmosis that you should learn. When will the passage, now the two solutions are separated by a semi-permeable membrane. And we have to understand, ये जो inward or outward movement कब बंद होगा? When will it stop? It will only stop when on either side of the semi-permeable membrane the concentration of the water is the same. It will only stop when the tonicity, or even if you don't go to the word tonicity, we haven't taught you so far. It will only stop when the concentration of the solution on either side of the semi-permeable membrane is the same. Okay? So beaker and funnel लिखने की भी जरूरत नहीं है. You have to judge by the membrane. Membrane के दोनों तरफ जब concentration same हो जाएगा, then the movement will actually stop. But generally, such type of a situation in reality does not happen. No, this is we are only talking about the experimental condition. Done? ये लोग तो पूरा पेपर लिख रहे हो आपके. हाँ? फाइनल पेपर है. फाइनल पेपर है. कितना लिखोगे? Let me check what you are writing. ज़्यादा नहीं लिखना है. ले. Number 1. What is number 1? ये कौन सा पेपर है? अरे ये कौन सा वर्ड है? सेलोफेन लिखने वाले थे तुम. सेलोफेन लिखने वाले थे तो सेलोफेन का स्पेलिंग क्या है? CELLOPHANE. ठीक है. अच्छा. What will happen to the liquid in the beaker? Liquid in the beaker मैंने पूछा है. Funnel. What will happen to the liquid in the funnel? Beaker में अगर hypotonic solution है, the liquid in the funnel the level will rise. Solution क्या? Okay. Number 3. Reason क्या है? Number 3 का आंसर क्या है? Reason क्या है?
Funnel is concentrated and beaker is diluted.
हाँ.
So water will goes from diluted to concentrated.
Very good. Water will move from a dilute to a concentrated medium. स्पेलिंग्स पे ध्यान दो, स्पेलिंग मिस्टेक बिल्कुल नहीं होना चाहिए. तो constant concentrated medium. Okay. Next is what? Will there be any change in the taste of the water? That is number 4. No. Number 4 में answer is no. हाँ, which one? This one? Yes. There will be a change in the yes. हाँ, तो तुमने पहले no लिखा था. Then you got confused. फिर तुमने yes लिखा. और अगर मैं तुम्हारा नोटबुक नहीं खींचती, फिर तुम कंफ्यूज हो जाते, इसको काटते, फिर वापस तुम लिखते. Why there is a confusion? Repeatedly मैं बोल रही हूँ. Confusion है, पूछो. कहाँ पे confusion है? Clarify your doubts. अभी समझे? इंग्लिश एंड हिंदी यही लैंग्वेज मुझे आती है. मेरा मदर टंग तुमको नहीं समझेगा, तुम्हारा मदर टंग मुझे आता नहीं है. सो यही दो लैंग्वेज है मैं तुमको समझा सकती हूँ. प्लीज आस्क क्वेश्चंस यही दो लैंग्वेज में, देन आई कैन एक्सप्लेन प्रॉपर्ली. बट डाउट्स में बार-बार पूछ रही हूँ, यू आर स्टक टू व्हाट यू आर राइटिंग, कैंसिलिंग एंड राइटिंग. अरे पूछो, क्लैरिफाई करो. हमेशा जो भी इस तरह के एक्सपेरिमेंट बेस्ड क्वेश्चंस आएगा, when you read you are able to understand बीकर में क्या है, फनल में क्या है, कौन सा मीडियम कॉन्सेंट्रेटेड है, पहले वो तुमको क्लियर हो जाना चाहिए. Now the rule is what? The water will always move from a dilute side से concentrated side में जाएगा. So यही रूल हर जगह तुमको अप्लाई करना है. You got confused with that part. Again I will explain. अगर if I am talking about 95% solution, अच्छा नहीं 95 नहीं, I am talking about 25% sugar solution and 5% sugar solution. Which one is dilute? 20 funnel छोड़ो. 25% sugar solution and 5% sugar solution. Which one is dilute? 5%. आया समझ में अभी? 5% मतलब 95 parts water है. Clear है ना? So ये सारे concepts अगर clear है, then you are sure कि ये वाले answers full 5 तुमको मिलेगा. And don't give pain to our eyes so much. Practice करो, write. As you read also write a lot. Handwriting थोड़ा सा legible करो. बोलो. Done? You are done? You write all the answers now?
Now I did not get the second one.
कौन सा second है? What will happen to the have to understand? What will happen? The solution in the funnel will rise. Solution in the funnel will rise. Done? करो. I just last 1 minute करके फिर करो, complete करो, देखो. Done? Now one more experiment we are going to learn. One more concept we are going to learn here. So I hope this type of experiments, any type of a question comes, any type of a change, what is the first thing that you should make clear for you before you answer the questions? Which medium is concentrated, which medium is dilute? And वो जो membrane कौन सा दिया हुआ है? Is the membrane semi-permeable? अब यहाँ पे अगर मैं semi-permeable की जगह अगर मसलिन देती, तो the entire set of answers would have been different. तो ये तीन चीज पहले चेक करना. Which medium is concentrated, which medium is dilute, and what is the type of the membrane being shown in the figure. ये तीन चीज पे ही तुम्हारा सारे answers depend करेगा. Okay. Now let us learn one more concept. What is it? अभी यहाँ पे a situation is something like this where the beaker has water. Yes, the funnel has a solution, sugar solution, and this is the initial level of the sugar solution, and this is the initial level of the water. This is the initial level of the water in the beaker. So now, when I talk about the initial level of the solution in the beaker, of the water in the beaker, and initial level of the sugar solution in the funnel, what type of a membrane I am taking? Semi-permeable. compartment, cellophane. Other examples can be the bladder, okay, the bladder membrane, the goat liver, or the goat bladder, or these are examples of semi-permeable. Now, in this experiment, a little bit of a change is, with the same setup, what do we take is, we take a piston. We take a piston and we attach this piston. The piston needs to be attached also. We need to hold this with the help of the clamp and the stand. That's different. So now we are taking a piston here. What is my piston? Something like those pitch carriers that we have. No? So similar that way, those type of setups are also there in the laboratory where we can attach a piston and keep some weight over it. Keep some weight over it. Now we keep it for some particular period of time. We placed a piston inside in the funnel, almost touching the level of the solution, and we kept some weights over it. Wait for this particular period of time. Now what do we see? After this particular period of time, we get to see that ये जो level of water in the beaker, no, it's not showing much change. It's not showing any remarkable change. What about the solution in the funnel? Is it showing any change? No, that is also not showing any remarkable change. So both ways, there is hardly any change. But setup तो पहले जैसे ही है, क्यों कोई change नहीं है? Change तो होना चाहिए था. Now the change is not there because initially, as you start with the setup, initially few molecules, very few, may enter, but after some time or just after a few seconds to minutes, couple of minutes के बाद, since we have already placed the piston and the weight, this is going to exert some pressure on the membrane. Something like, if I take the water filled up in the pitchkari and then when I push it with the help of the piston, what's happening? There's enormous force on the nozzle. The water is being pushed in the nozzle and that's the reason the water is sprinkling. So यहाँ पे तो कोई holes नहीं है. That's the reason the water is not able to sprinkle, but there is a pressure generated. So there is a pressure which is generated on this side of the semi-permeable membrane, जिसकी वजह से the water is not able to enter. Initially, few molecules must have entered. उसके बाद बंद हो गया. The more and more this pressure is exerted, the movement has stopped. Even though this medium is hypotonic and this medium is hypertonic. अभी भी तो ये medium hypotonic है, ये hypertonic है. But now there is no movement because there is a force exerted, pressure exerted on this side of the membrane, and इसके वजह से the movement from this side has stopped. What is this called? This is what is known as osmotic pressure. This is what is known as osmotic pressure. What is the definition of osmotic pressure? The minimum pressure which prevents, this pressure is preventing the movement of molecules this side, no? The minimum pressure which is preventing the movement of molecules of the solvent. What is a solvent, no? Movement of the solvent into a solution. ये movement को रोक रहा है ना? What is osmotic pressure? I repeat again, the minimum force, the minimum pressure which is not allowing the movement of the solvent molecules into the solution. This is solvent, but this is a solution, whatever dilute solution or concentrated solution we are talking about. अब इस experiment में we are not talking about the concentrated and the degree of concentration, but यहाँ पे we are only interested to know, this is of course dilute and this is concentrated. What is the degree of concentration? वो नहीं चाहिए. But यहाँ पे तो movement इस तरह से होना चाहिए था, but क्यों नहीं हो रहा? Why not? Only because there is a force exerting on the other side. Same like that. 5 of you are pushing this door with all your force. The other 5 pushing from the other side. Are you able to go out? No. Are they able to come in? No, because this is acting as a semi-permeable membrane. You are also pushing, trying to enter. This is also trying to enter. But since they are also pushing from the other side, that's the reason you are not going able to enter. No, the same thing. This is they are pushing so hard from out that side, you are not able to go out. The pressure is big this side, as a reason this force is not acting at all. Even though there is a concentration difference, it's not working. This type of a pressure which does not allow the movement of a solvent into a solution, which always normally happens. Solvent molecules always enter into a solution. क्योंकि solvent medium हमेशा dilute रहता है. The minimum pressure which prevents the movement of the solvent molecules into a solution is always known as osmotic pressure. Osmotic pressure. So what we have completed is with the concept of the different experiments on osmosis, osmotic pressure. We have also talked about the concept of the different concentrations. What are those terms? We had already learned about the concept of hypotonic solution, hypertonic solution, isotonic solution. We had also talked about conditions when we understand that what should be the degree of movement of the molecules inside the animal cells, plant cells. Now look into this and try to answer these questions. Take one question. You may refer to this diagram. I already explained you this. Take one question, write down. In a solution of specific strength, certain RBCs are placed. Full stop.
What can I repeat?
In a solution of, and write down the question you can write down. When you answer, read properly. यहाँ पे technical term use नहीं करना है. Technical term यहाँ पे we should not repetition, it should not be. यहाँ पे भी इसका आंसर यहाँ पे लिख दिया, यहाँ पे से नहीं, ऐसा नहीं. You should know how to write. यहाँ पे वो technical term खाली use होगा. Don't take too long, 5 minutes should be enough. फटाफट complete करो. Done. Over. All done. Here you go. What's your name?
Sohan.
Sohan.
Han.
Sohan. In your school, they have finished with
Absorption by roots, transpiration and photosynthesis.
endocrine glands and excretory system, no?
No.
होता ना?
Yes.
अच्छा, हाँ होता है.
answer in this field.
Are you able to answer? Next. In a solution of specific strength, certain RBCs are placed. Let me draw, tell you what's happening. In a solution, I have taken a solution, I don't know what is the strength. I don't know what is the strength. And I have placed certain RBCs here. But after 8 hours, no change. RBCs are as it is. RBCs are as it is. Give a reason for this observation. After 8 hours, no change. Give a reason for this observation. Concentration here and concentration here must be the same. No? Answer is concentration of the solution and the RBC is the same. That is the answer for the first one. A. Concentration for solution and RBC is the same. Or concentration of the fluid, you can write concentration of the fluid in the RBC and concentration of the fluid of the or concentration of the solution and RBC is same. That's the easiest way to write. Number B. Technical term क्या लिखेंगे? What is the technical term that you can use for this solution? Isotonic. Third, what changes can be made in the solution to make the RBC swell? Now I want the RBCs to really swell up. तो swell होने के लिए water should enter. Water कब enter होगा when this is not of the same concentration but ये dilute होना चाहिए. So I will add water here. तो जैसे ही मैं water add करती हूँ ये solution अभी dilute हो जाएगा. If I am adding 2 cups of water, so जो भी solute है ये dilute हो जा रहा है. RBC तो same रहेगा concentration. So now this is becoming a dilute medium. अब जब ये dilute हो गया, now slowly water will start entering, and after some time I can see the RBC has started swelling up. The RBCs have started swelling up. So that is the C. What changes can be made in the solution? Add water and make it dilute. Make dilute by adding water. Achha, D. What process is expected in the RBC now? Inward movement of water. What is it? Endosmosis. So a condition, now the RBC is filled with water, no? RBC is gradually becoming tight filled with water. So a plant cell which is also tight filled with water, what is this condition called? Turgid. Did you write turgidity? Turgid. Turgidity is the phenomenon. Condition I asked you. What is a similar condition in the plant cell known as when the cell is filled with water? Turgid. Turgid. Clear है? Are you clear about the concepts? कोई doubt है? कोई doubt है? Today itself we will go and read this part of the chapter ताकि जो भी confusion है clear हो जाए, so that when we discuss the next parts of the chapter you should be very clear. Now homework you should be done with these questions. Now check करो. Number 2, number 3, page number 53 का number 2, number 3 should be done. Number 5 also. We have already finished this concept. Number 2, 3, 5 should be done. Okay? So this will remain as homework for you. Done? Anything to clarify? Complete this part of the chapter. Read. Complete these questions. कुछ भी doubt है, ask me next class. Once you read, I'm sure you will be having some doubts, confusion. So it's better don't wait for an exam. Try to complete it now itself.
इसमें आए मैंने आता पर तू आया फाइनल एग्जाम ऑफलाइन ही पास डॉक्टर सब ऑनलाइन ही हमारा टाइम फाइनल ऑफलाइन पता नहीं हमारे स्कूल में